Okay, before moving on with fermentation, I wanted to clear up a few things that weren't perfectly presented in the last video. Although I made annotations in the video, I'm going to go ahead and clean it up in here, and then talk about something else real quick before carrying on with fermentation. So for this one, I'm going to go ahead and erase this and put ATP to ADP. And for this one, we have also ATP to ADP. And on this last one, phosphoenol pyruvate, or PEP, going to pyruvate, we have ADP going to ATP. Now, just to be clear, this way of writing out reactions is commonly done when you have an activated carrier either being used or it's being activated. So what do I mean by that? So if we go up here, for example, fructose 6-phosphate to fructose 1,6-bisphosphate. In this case, an ATP is being used. So an activated carrier is being used to drive the reaction. If I were going to write out this reaction completely, what I would have is fructose 6-phosphate plus ATP going to fructose 1,6-bisphosphate plus ADP. Now, if you're going to look at this reaction, we can actually go to what we had above for coupled reactions. So let's just go way back. Right here. This thing, let me actually erase all this stuff. If you look at this, you can think of A being fructose 6-phosphate. So instead of having AOH, I could just put fructose 6-phosphate. And we want to get to fructose 1,6-bisphosphate. But adding an inorganic phosphate by itself would have a delta G that is larger than 0, so it's unfavorable. But if you couple it to ATP hydrolysis, right here, then what you get is a reaction that is actually favorable, right? Because ATP hydrolysis is very favorable. So you can think that, okay, here I have fructose 6-phosphate plus inorganic phosphate plus ATP plus H2O going to fructose 1,6-bisphosphate plus H2O plus ADP plus PI, and then I get rid of like terms on both sides. 2 PIs here, 2 H2Os here. What am I left with? With the fructose 6-phosphate plus ATP going to fructose 1,6-bisphosphate plus ADP. Right? So let's go back to where we're at. All right, so whenever there is something like this, usually that is an activated carrier being used, or in this case here, the activated carrier is being, well, activated, right? Because ADP is going to ATP, so this is an activation step of the activated carrier. What I wanted to talk about also, here I have written out glycolysis. I have hidden all the reactions that are termed as neither favorable nor unfavorable, meaning that at cellular concentrations, those reactions can sort of go either direction, meaning that their equilibrium constant K is really close to 1, so if the concentration of the product of that reaction increases a bit, the reaction will start to move in reverse, whereas if the amount of reactant starts to increase compared to the product, it will start to move in the forward direction. That's what we mean by neither favorable nor unfavorable. It doesn't mean that the reaction itself doesn't have a delta G standard or equilibrium constant. It just means that that delta G standard is relatively close to 0 and the equilibrium constant is pretty close to 1, and changes in the concentration of the reactant compared to the product and vice versa could shift the reaction to favor the forward or the reverse direction. That's what that means. So those steps are being hidden by these big green arrows. So wherever you see the green arrows, right here, for example, and here, it's basically telling you that there are reactions that are being hidden. But these are the reactions that are, of course, important to glycolysis. We're hiding them because we want to look at something very specific, and that is the regulation of glycolysis. So I'm going to go ahead and erase these. All right. So we know that there is negative feedback loop on the first favorable reaction where glucose 6-phosphate inhibits hexokinase. And we also know that ATP will inhibit both PFK and pyruvate kinase, PK. Now the thing to remember, it's something we talked about before. Although we say it is inactive or active, that's not really true, because if we say inactive or active, we mean that it's either on or off, and that's never the case. Because your cells always need ATP. We are a low entropy being. All life is low entropy. And to maintain this low entropy being, there has to be a constant input of energy. Now, prior, this was something that was sort of, what is energy? What are we talking about here? But we kind of know now, right? Because if you want to move towards lower entropy, meaning more order, that is unfavorable. Thus, you have to have reactions coupled to these processes that organize things that are actually increasing entropy more. And that's where this comes from. This energy thing that you probably heard of many times before. So where does this energy come from? It mostly, if not all, comes from ATP. Whether directly being like, for example, glucose to glucose 6-phosphate, where ATP hydrolysis is directly coupled to that reaction, or indirectly. Now, the indirect coupling of ATP hydrolysis to certain processes is something that we will see an example of in section 2. So let's not worry about that right now. So the cell always needs ATP, even if you're resting, doing nothing, just to maintain the structure of life, the structure of the cell, the structure of the tissue, the structure of the different tissues in relation to each other in the organism. That organization will require the use of energy, the coupling of ATP hydrolysis and other activated carriers to processes that lead to that organization. The only process that gives you that energy, meaning it breaks down the food products that you eat to give you ATP, is cellular respiration. So you cannot shut off cellular respiration. If you shut off cellular respiration completely to 0%, and remembering that cellular respiration starts at glycolysis, then the organization will start to fall apart and the cells will die, which leads to the tissue decaying, which leads to the organism dying. Okay, so we cannot shut off glycolysis completely. But as you can see, as ATP concentrations go up, you start to have inhibition on PK and PFK. And as that happens, the amount of glucose 6-phosphate will increase because the reactions that are here will reach equilibrium and the amount of glucose 6-phosphate will start to build up. And then you have inhibition on HK, hexokinase, which leads to glucose building up in the cell, which is a good thing because that's what we talked about before. If glucose builds up in the cell, less glucose will diffuse into the cell from the extracellular side. Thus, that extracellular glucose can move on to cells that actually need it through the circulatory system and etc. Now, what does it actually look like? So, I have a little graph here that we're going to look at. First, we're going to say, okay, the enzyme HK, or hexokinase, which is the enzyme right here, right? What does this activity look like? Well, it's going to seesaw back and forth, right? Because if it starts off near 100%, then as the amount of glucose 6-phosphate goes up, its activity starts to go down, right? But then the cell needs glucose 6-phosphate to be processed through glycolysis to make pyruvate, and the pyruvate used by the citric acid cycle to make the products of the citric acid cycle, which then some of those products, specifically NADH, will be used by oxidative phosphorylation to make more ATP. So as the activity of hexokinase goes down and glucose 6-phosphate levels are sort of stabilizing, the glucose 6-phosphate is then going to be used by the rest of glycolysis, right, going in this direction. And glucose 6-phosphate concentration starts to go down, but if glucose 6-phosphate concentrations start to go down, the inhibition that we see here starts to be lifted slowly. Not 100%, but slowly be lifted. And thus, this will start to swing back up, right? More glucose 6-phosphate is being made. But as more glucose 6-phosphate is being made, well, then the inhibition on hexokinase increases, so it starts to go down, right? As you can see, this is sort of a sinusoidal wave that goes back and forth between a certain range. It never really hits zero. It kind of might get close to 100%, but it probably never will. And it sort of stays in a range, which I do not actually know what this exact range is, but it's going to stay within a narrow range of going back and forth, thus stabilizing the amount of glucose 6-phosphate in the cell at any time. Right? And just to make sure, the x-axis is time. Now, with the other ones, you can think of it in the same way. It doesn't matter. It could be HK, it could be PFK, it could be pyruvate kinase. Now, how do you go from 100% or somewhere below 100% to maybe 50% activity and etc. when you have an enzyme? Well, remember what we said back when we were talking about enzymes initially. We're not talking about a single molecule of enzyme. We're talking about a population of that enzyme. So there's a certain concentration of hexokinase in the cell. Yes, a single hexokinase is either completely on or completely off. But if you have 80% hexokinase activity, that means 80% of hexokinase molecules are active while 20% are inactive. Okay, so that's all I really wanted to talk about. Talk to you guys later. Bye-bye.